get started now, the intro of our early stage investigator. So Gina Borgo and Rachel Rudishauser's lab. We're delighted to have her here, give the ESI talk of the day. Her work focuses on utilizing the rhesus macaque SIV model to understand what qualities in the antiviral CD8 T cell response contribute to the control of virus replication during analytic treatment interruptions. She received her PhD at UC Berkeley in infectious diseases and immunity, where she worked in the laboratory of Matt Welch to describe the role of bacterial phospholipase in the intracellular life cycle of rickettsia. And previously, she worked with Jane Kaler on Bartonella quintana and the transition from the body labs to the human host. So Gina is passionate about scientific communication and training the next generation of scientists. In the future, she hopes to apply her research experience to help develop accessible therapeutics for challenging pathogens. So we're delighted for you to give a talk today. Gina, please take it away.
All right. Thank you, Peter, for that great introduction. And thank you to the organizers for inviting me to speak today. Okay, I think we are good. Okay. So as Peter mentioned, I am a postdoc in Rachel Rudishauser's lab, and I'm interested in understanding what makes a really good antiviral CD8 T cell. And I'll tell you a little bit about the project that I've been working on in collaboration with our speaker today, Dr. Atha Mukwege, trying to understand ideal CD8 T cell responses. So currently, lifetime antiretrovirals, or ART, are required for most people living with HIV due to the persistence of the HIV reservoir. And so I have here a representation of an HIV plasma viral load graph with the typical dynamics during acute infection, once on ART, when virus is suppressed. And then most relevant to my talk is this very rapid rise in the plasma viral load if ART is stopped, what we refer to as viral rebound. And so what this is meant to really demonstrate is that even after being on ART, that the immune system is unable to respond effectively to prevent this rebound, or even to sort of alter the set point in most circumstances. And so when thinking about how to suppress the virus without ART, or how to design therapeutics to suppress the virus in the absence of ART, we really want to understand how we might modulate the immune system to change these dynamics once ART is stopped. And so in the Rudishauser lab, we have both human and NHP cohorts, and we really want to understand that during what is happening during analytical treatment interruption, or ATI, when ART is stopped, to understand immune features that might be associated with HIV or SIV control. And so this is the same plasma viral load graph that I had previously, and it's depicting sort of two classifications of post-treatment interruption or off-ART outcomes. The teal graph is high plasma viral loads post-ART, so they rebound and then they go back to a similar set point as previously. And this is in comparison to this orange graph, where a participant or an animal has a lower plasma viral load set point than initially. And so we really wanted to understand that by comparing these two outcomes, what are the differences in the immune features, or what's happening in the immune system, to better understand how the virus is interacting with the immune system. I also want to point out that we have particular squares at time points on ART and early off of ART. And these are time points that we're really interested in and think we can extract a lot of interesting and useful information, because the differences that we would observe here are less explained by differences in plasma viral load, where we know we should expect some differences in the qualities or the features of the T cells, or of the immune cells. And so we are specifically interested in CD8 T cells because they're good at killing virally infected cells. And I will tell you today about the non-human primate cohort that we've been using to try to understand CD8 T cells during treatment interruption. And so what evidence was there to support CD8 T cells playing a role in controlling virus replication off ART? So Dr. Okoye had previously published some very intriguing results, and some of the earliest results that CD8 T cells might play a role during treatment interruption. So this is showing you SIV RNA levels when ART is stopped. And in red are animals that had CD8 T cells depleted prior to and during the early stages of the treatment interruption. And as you can see, that compared to control animals or animals that had less efficient CD8 depletion, there's a 2-log difference in the set point. So it didn't affect time to rebound, but not having T cells meant that you ended up at a higher plasma viral load set point. And so this was intriguing. And then in addition, Opham had also found that within this control group, there is a range of plasma viral load outcomes when the animals come off of ART. And so they're all infected with the same SIV, all on ART at the same time, taken off at the same time, but then end up at these different set points. So we really wanted to understand what is the difference in the CD8 T cells between low plasma viral load set points in yellow and orange, and these higher ones in blue and teal. And some qualities that we think might be important are really based on literature on CD8 T cells from elite controllers. And this includes things like high proliferative capacity, high expression of a transcription factor that promotes memory-like proliferation capacity called TCF1, higher frequency in sites of SIV infected cells, such as the lymph node and the B cell follicle, and then the ability to create secondary effector cells and maintain an antiviral response. And so in order to answer this question and really understand if animals with these lower plasma viral load set points all have less exhausted, more stem-like CD8 T cells, so higher proliferation, we took the animals from Opham's control group, and at pre-ATI and the earliest time points post-ATI, about a week and 2 weeks, we wanted to look at the immunophenotypes of the CD8 T cells between these two groups. And just to show you what the plasma viral loads look like at those time points, all of these animals are virally suppressed on ART pre-ATI. You start to see some differences in the plasma viral load a week post-ATI, but it's not starting to segregate by where the animals end up in terms of set point. But by day 15, the animals, the plasma viral load is very high, and the animals have started to segregate by where they'll end up at set point. And so to immunophenotype these CD8 T cells, we looked at the different CD8 T cell subsets, effector function, exhaustion, in vivo activation, and in vivo proliferation using Ki-67, which I'll talk to you about now. So animals with low plasma viral load set points, we did find had a higher frequency of Ki-67 CD8 T cells in certain memory subsets pre-ATI, so when all of the plasma viral loads are very similar and undetectable. So here are different CD8 T cell subsets, stem cell memory, central memory, transitional memory, and effector memory. And I have sort of their continuum of properties underneath. And as you can see in the central memory and the transitional memory, you have a higher frequency of cycling CD8 T cells in the central memory and transitional memory. And there also seems to be a trend towards the frequency of cycling transitional memory at pre-ATI and where the animals end up in terms of set point. I also mentioned that being well positioned to where there are where the SIV infected cells are in the B cell follicle might be an advantage. So here you have infected follicular T helper cells, and then these green are HIV specific, and the expression of CXCR5 is what allows these CD8 T cells to traffic into the B cell follicle. So we think that this might be important for positioning CD8 T cells well. So again, if we look at cycling of the memory of these CXCR5 positive CD8 T cells, you can see that specifically pre-ATI, we see a lot more cycling in the animals that end up at lower plasma viral load set points. And the correlation with the post-ATI set point also trends towards a negative correlation between that cycling follicular and the set point. So just very briefly, one slide on what's happening post-ATI. We were interested in TCF1 because, as I told you, this seems to promote memory-like proliferation capacity and has been shown to be important for elite controllers and correlates with expansion capacity. So we looked at the percent of TCF1 positive in the Ki67 populations within all the memory subsets at day 15 post-ATI. And as you can see, the transitional memory specifically are really maintaining a higher expression of Ki67, of TCF1 in the Ki67 population. And this correlates really well with the post-ATI set point. Of course, the plasma viral loads start to diverge, and TCF1 can be, its expression is correlated with antigen levels. So I wanted to just show that we also observe this negative correlation between TCF1 in the transitional memory that are cycling, also at pre-ATI. So this seems to be there at baseline, which is very intriguing. So what I've shown you here today is that pre-ATI, there were some features that were observed consistently with the lower plasma viral load set point animals, and that included cycling in memory CD8 T cells, in these follicular CD8 T cells, and they tended to trend towards higher cycling pretty much across the time points, although it wasn't always significant. We also saw higher TCF1 frequency in the Ki67 population. So we think this suggests that the animals that start out and continue to maintain this larger fraction of cycling CD8 T cells, especially in the lymph node, might be more likely to control the plasma viral load long term. So this might have implications for proliferative potential as being a good target for immunotherapies. And so with that, I'd like to acknowledge the lab, of course, Dr. Okoye and his lab, and everyone who's helped with this project. And now I can take questions.
That's great, Gina. We'll open it up for questions. I can start with one. It looked like you were describing mostly the bulk populations of CD8 T cells. Are you also planning on doing studies of the SIV-specific CD8 T cells?
Yes. So we had class 1 multimers for 2 immunodominant responses within these macaques. And we didn't observe the same trends when looking at that. Seemed to correlate more with plasma viral load than it did with sort of what I've shown here, where it's not easily explained by antigen burden or plasma viral load. So there were some differences, mostly post-ATI, but I do not observe the same cycling phenotype pre-ATI within the SIV specific. But we plan to dig in more to try to figure out what's going on with the SIV specific and still have more experiments planned to drill down on the SIV specific population.
Okay. Well, just to say, Gina, so those epitope specific responses may not be representative of all the responses. So you would probably need to look at more for variety or something.
Yeah.
Other questions from others? Steve?
So great, Gina. Probably know my question. I always ask the same question. So how might we begin to think about developing this as a potential biomarker, surrogate marker, whatever you want to call it, as a way to sort of begin to do drug discovery, vaccine discovery for interventions that can induce control? Is this the pathway you think that we're going to need to go with immunotherapy? And if so, is this a measurement that we could be using outcomes, and what kind of therapies you think could generate this type of response?
I think it might be part of the puzzle. I think confirming these results with additional assays might be helpful, or understanding how it might relate to what type of response you get if you have a lot of these cycling cells pre-ATI. I think that'll be an important part of the puzzle. And so I think, in short, both for time and just brain fart, is I don't know. I think it's going to be part of the puzzle and the question. And this sort of suggests that you might need to increase this proliferative capacity prior to the treatment interruption and try to get, the SIVs, maybe the SIV specific, maybe this has to do with the general immune environment. And so there are so many questions. It's difficult to say exactly what this means, but it definitely suggests that, at baseline, you really know, like there might be really good indicators of what will happen, and therefore those changes to the T cells that you might want to make need to happen on ART or pre-ATI.
Hmm.
So that's, I
Maybe-
think.
Yeah, no, yeah, great. Maybe often we'll dive a bit deeper into that specific question as it pertains to predictors of outcome in his vaccine studies.
Yes,
How's that for a segue?
there we go. Okay, so
Gina, do you want to take it away with your introduction of Afam?
yes, so it is my great pleasure to introduce Dr. Afam Okoye. He is a professor at the Vaccine and Gene Therapy Institute and the Department of Molecular Microbiology and Immunology at Oregon Health and Science University, with a joint appointment at the Oregon National Primate Research Center. So Dr. Okoye received his PhD from the University of Glasgow, studying papillomavirus capsid proteins, and he moved to Oregon Health and Science University for postdoctoral studies with Louis Picker and joined the faculty in 2013. I think I'm most struck by his impressive range of publications that are really defined by his ability to utilize the rhesus macaque model to answer key questions about how therapeutics affect both the virus and the immune system and what these perturbations do. So his research currently involves utilizing NHP models to understand mechanisms of HIV persistence and to develop therapeutic interventions aimed at achieving durable remission. His work has elucidated details on reservoir establishment and dynamics on ART, the role of different CD4 subsets during progressive SIV infection, and the role of CD8 T cells during treatment interruption. He's also been instrumental to answering key hypotheses around the effects of latency reversal agents, cytokine agonists, broadly neutralizing antibodies, B-cell follicle disruption, and therapeutic vaccination for T-cell responses. And I think his work really speaks to his commitment to solve really big problems, but in really practical and broadly informative ways. So, of course, I had the pleasure of continuing to work with him on this project that I just told you about, but I will say that I think Afam has a lot of mentees. I see people talking to him all the time at conferences, and I think it's because he's so generous with his time and advice and expertise. And most of all, he's one of the most, maybe this reflects my interactions with him, easygoing people I've ever met. He always just makes you feel really supported and capable, and like anything is possible. And so I'm honored to welcome Dr. Okoye today for his talk, Enhancing CD8 T Cells to Facilitate the Immune Interceptive Rebounding HIV and SIV Reservoirs. So please join me in welcoming Afam Okoye.
Wow. Thanks, Gina. I wish I was recorded so I can show it to my kids.
It's recorded. We have it
That was quite an introduction. Thank you. And it's really an honor to be here. And so thank you so much for the invitation. And I'm just, I know so many of you already, so it's really fun to continue our discussion. So thank you for this. And I will start, and I don't have to spend time with this, but we know that HIV is still important. So we're all here because we're trying to find the solution to this great burden of HIV. And again, I don't think I need to convince anybody here that we need a cure for so many reasons, which continues to be an issue today. But toxicity, cost, access, stigma, these are all really important reasons why my group and so many others here also are really interested in this idea of can we cure HIV. And I really hope that this momentum, the political and will to keep pushing towards a cure continues, because I think it's really important that we find a solution for HIV in our generation, hopefully. But anyway, so, one of the things that keeps me going is the idea that we know that, a cure is really possible. And, these are rare cases, but, like I always keep saying, like what we do in NHP studies, we're always looking for signals, because when you see a signal, you can bounce on it. And that's why I love Gina's data so much, because, it's all about signals. When you see a signal, then the question is, what does that mean? And how can we bring that out to kind of continue to push the field forward to understand how we can drive immune control? And these cases of people who have been cured of HIV, to me, continue, and the more cases we begin to hear, to me, always gives me the motivation to keep going, because we know a cure is possible. Now, obviously, here the cure was driven by allogeneic stem cell transplantation in at least 5 individuals. But the 2 people that did not get allogeneic stem cell transplantation that we believe have been cured, which Lorraine and the Esperanza patient, really talks to the fact that indeed the immune system potentially has a role in driving a cure. And so that's why I think, continue to understand how we can re-engineer or educate the immune system to provide a cure for HIV might be an important way to move forward. Just to highlight recently that, the question is about whether CCR5 is important for the cures that we have observed so far. New cases are showing that perhaps maybe not. And then this also, again, I think it's all about signals. We always thought that you have to have CCR5 delta 32 resistance to be able to achieve cure with allogeneic stem cell transplantation. But now we have these new patients, both the Geneva and the Berlin patient that was discussed this year at IAS in Munich, suggesting that perhaps you may not actually need that, and there might be signals within the whole process of transplantation, graft versus host, whatever, that actually might mean that we can find a way to actually eradicate nucleic acid in people over time. So this is kind of really exciting to me, and I'm looking forward to more of these cases coming up, because I think the field is then growing and gives us ideas of how we can move the cure agenda forward. But anyway, going back to what I was going to be focusing on today, because we know that in most cases, as Gina has really nicely described, that art alone is not a cure. And in most of the time when people who are living with HIV go off therapy, there is ongoing virus replication, and in most cases, there's progressive progressive disease. We do have rare instances of individuals who can control the virus by they have low levels of plasma viral replication, and we refer to these as elite controllers. But these are rare, and the question is, how can we drive people to move from the becoming progressors to more of elite controllers, or complete, which have no evidence of plasma in the blood, but potentially the evidence of the virus still exists. But the challenge for this is the fact that we have latently infected cells in many different sites in many different tissues. And, unlike the tumor setting, where I think, the challenge is probably a little bit easier for them when you have, a tumor in one location that you can target. Here we're really expecting the immune system to really do wonders, right? Targeting all of these sites where there might be potentially latently infected cells in all different tissues, and all these different tissues have a whole bunch of different immune microenvironments that govern the way the immune system operates in those sites. And so, to be able to drive a systemic control of viremia in multiple different sites, multiple different tissues, is a challenge. And so that's, as I just said, what happens is that you stop ART, the virus is reactivating. You have these multi-foci of infection, as opposed to what you would expect during primary infection, where you're getting just one site, potentially one area where there's initial founder viral spread. But here, in the context of therapy and off-art release, you're seeing the virus, potentially reactivating multiple different sites. So the immune response and the T cells that we are hoping would clear these reactivating infections need to be able to intercept in these different sites and these different places. And so that obviously is a challenge. So, one of the first things we tried to do to answer this question, and I might not spend too much time on it because Gina did such a great job of explaining the experiment, but the idea really is, we need to start from the beginning and ask, what do CD8 T cells do at the time we stop ART? And I think that's one of the things I really loved about this experiment, because it just began to give us the baseline to begin to think about the future. Right? If you understand their limitation, you can think about, okay, this is where their limitation is. What can we do to boost that, to be able to drive control of the virus going forward? And that's why we specifically in this study chose animals that we know will be predisposed to control the virus. So they had an MHC allele that allowed that, usually, as enriched in controllers. And then we depleted them of their CD8 T cells just prior to ATI, and made sure that it was steady state depletion even through ATI out release. And then we looked at the dynamics of the virus. How does the virus reactivate and rebound, and what's the nature of the rebound when you have no CD8 T cells that should intercept that rebounding virus? And so in this context, we can see that animals that had were fully depleted of their T cells, in both in the blood and the lymph nodes, we can see that all of them rebounded. And set point was higher, almost 2 logs higher than what happened when you had either an intact CD8 T cell compartment or incomplete CD8 T cells. But most importantly is the fact that, when you look very early on to see at least the rate at which rebound is occurring, the time to rebound, it was surprising to us that we didn't really see much of a difference, in terms of the initial early dynamics of how the virus rebounds. And so it tells us that, CD8 T cells are important because they're important. If they're there, you have a robust CD8 T cell compartment, you can definitely control the virus a little bit better after R is released. But at least that early time when the virus is beginning to rebound, we don't see CD8 T cells making much of an impact. And this is probably because the CD8 T cells need to get activated. You get expansion of those CD8 T cells, and then they become effective. They become upregulate effector molecules and become cytotoxic, and then can begin to intercept the rebounding virus. But that rebounding is the activation is occurring when in response to antigen as the virus is replicating. So this really tells us that, in the absence of therapy, post-antiviral activity still follows viral outgrowth. So as just summarizing that whole study, you stop ART, the virus replicates. As it replicates, the CD8 T cell responses then begin to be activated. As they become activated, then they begin to challenge that reactivating virus, and then you get some control where set point is achieved. And so in this context, it's telling us that, the CD8 T cells are really too little and too late, which is a phrase that was coined many, many years ago by Ashley Haas. And so the question we've been asking for the last couple of years is really, how can we enhance the antiviral activity of CD8 T cells so that we can drive a further earlier immune intercept of the virus? And so with the idea that if you can intercept the virus that's reactivating much earlier on, potentially this could lead to more post-treatment control. So to get the CD8 T cells ahead of the rebounding or reactivating virus. And so, we've tried a variety of things. One of the first things we wanted to ask is what happens if you give immune checkpoint blockade, and we know that immune checkpoint blockade has been very effective, especially in the oncology setting and enhancing cellular immunity. And it's really been a game changer, I think. It's a huge field right now in developing immune checkpoint blockers. There was a paper published many, many years ago by my good friend Vijay and Rama, where they had given PD-1 blockade to some viremic animals and actually showed in that setting that they saw an improvement in viral control. And so since then, we've had PD-1 antibodies that have been resynthesized. This has allowed us to test it in non-human primates. And we've done several studies with it. And some of the initial pilot studies we did with PD-1 or in combination with CTLA-4 blockade showed that for the most part, we see some improvement in viral control in animals that have good T cell responses that are naturally controlling the virus very well, controlling SIV to a certain degree. And I'm not sure the CTLA-4 data here, but for the most part, whether you have PD-1 or you give PD-1 in combination with CTLA-4, you can actually get further immunological control of the virus just by blocking this PD-1. And I would say that it's not clear whether the cells are exhausted and we're reversing exhaustion in these controllers. That's why they're controlling better. But one of the things we do see is that PD-1 is actually PD-1 blockade actually releases the brake on proliferation. And when you give PD-1 blockade, the cells proliferate much better. And we see that even in monkeys that are healthy, that are not infected, if you give PD-1 blockade, you see memory cells proliferating. So going back to Gina's talk, it could be one of the things we're seeing this improvement is because we're restoring the proliferative capacity of these virus-specific T cells, and that is driving this control. But this control is mostly observed when animals are controlling it already. So at viral loads that are about less than 10 to the 3 logs. If you have animals that have higher viral loads, we would refer to potentially their T cells are not as effective at controlling the virus naturally. So even if you're restoring the proliferative capacity of these T cells, or reversing some level of exhaustion, those T cells aren't effective anyway. And so, you don't get as much improvement as you would if you have animals that have really good cellular responses to begin with. But this actually allowed us to then ask the question about rebound and ask whether, if you block PD-1 blockade at the time of ART release, do you then begin to see an improvement in the ability of the virus, the T cells to control the virus? Will it help the T cells to attack the rebounding virus a little bit earlier? So in the first study we did, we asked this question with PD-1 blockade. Here what we did is we started PD-1 blockade. We infected animals, put them on ART. When they were fully suppressed, we then gave PD-1 blockade by itself, starting when animals are fully suppressed, and then we maintained PD-1 blockade through several weeks after ATI. We also had another group that we started PD-1 blockade just prior to ATI, and then we had another group of controls. So these were here. So there were 10 monkeys in each group. And one of the things we're really interested in this study initially was just to ask, was this enough to increase the intercept of the virus? So if we look at the dynamics of how the virus is reactivating from latency when you have PD-1 blockade there or not, we didn't see, at least with like blocking PD-1 signal, we didn't see that the virus was intercepting earlier. However, what we did see, which I think is really important and it really helps us to think about how we use PD-1 blockade going forward, is that, in animals that did have PD-1 blockade, whether we started it several weeks prior to ATI or at the time of ATI, that even as the virus began to replicate, we actually saw that those animals could control this rebounding virus much better. So we did, to a certain degree, enhance the functional activity of those T cells. Now, whether that was because we increased proliferation, now in the presence of PD-1 blockade, we do see high levels of T cell proliferation of both CD4 and CD8 cells. Or whether there was, some exhaustion that we were reversing, or whether we had increased, cytotoxicity, it's still unclear. But what we do see is that, PD-1 blockade during this period when after ATI actually drives down the virus much better, and you get better control early on of viral replication as long as PD-1 blockade was maintained. However, after we stopped PD-1 blockade, we eventually followed the animals, and we actually saw that after you stop PD-1 blockade, this control was not maintained long term. And that, and so it really tells us that, we were able to do this transiently, but it wasn't persistent. And so the question is now how can we drive a more persistent control of the virus, with PD-1 blockade? And I would probably talk about that a little bit later towards the end of how we're thinking. But the other thing we wanted to do then also was to ask, well, what happens if we try and slow down the rate of rebound of the virus by repeating our PD-1 blockade study, but this time adding a neutralizing antibody, with the goal that, perhaps the neutralizing antibodies will allow the virus to rebound a little bit more slowly. And as it's rebounding a little bit more slowly, we can then see if adding PD-1 blockade at that time allows the T cells to be more effective and control the virus much better. So, again, in this study, we put the animals on ART. And this time we just started PD-1 blockade just prior to ATI, and we maintained it for several weeks after ATI. Now, in one group, some animals got PD-1 blockade in combination with two broader SIV neutralizing antibodies, which is K11 and ITS-103. So the PD-1 blockade was started about a week before ATI, and then we gave the bNAb just at the time when we expect ART to wash out, which is about 3 days after ATI, and then we maintained PD-1 blockade. In another group, we just gave them the antibody by itself and then a control antibody, so no PD-1 blockade. So this group is just showing that what happens to what the antibody does naturally. And as you can actually see the antibody by giving a bNAb at the time of ART, you can substantially delay time to viral rebound. And this also occurred here as well in the PD-1 combination group that, given the bNAb at the time of ART, substantially delays time to rebound, which is kind of what we were hoping for. However, the goal was not to just delay time to rebound. Our goal was to use the antibody to slow down rebound, so to change the ramp up of the virus. But, from a lot of the studies we've seen so far, even after this, was showing that, the bNAbs were kind of behaving like ART. As soon as the bNAbs wash out, the virus replicates, and the dynamics of reactivation was very similar to what happens when you stop ART. So, it's very hard to change that dynamic of reactivation, rebound of the virus, either with ART or with the bNAb. However, when you combine the BNAb with the PD-1 again, we saw that animals were controlling the virus, much better. And, from peak, this decline from peak was much more dramatic in the animals that had PD-1 blockade. So PD-1 blockade is effective, and there is an antiviral effect that we are seeing with PD-1 blockade that happens during the time of treatment. For the most part, though, after treatment is stopped, we begin to lose that antiviral effect in most of the animals, and trying to understand why we're losing it and how we can sustain it is one of the questions we're really interested in asking as we go forward. But then, as I said, when we looked at time to rebound, if you had the BNAb or PD-1 or alone or with combination with PD-1, the time to rebound between these groups were not substantially different. However, as I said, we do see that the animals, when they're in the presence of PD-1, they does increase and improve the antiviral effect of leading to lower viral loads early on. But this is not sustained long term as soon as we take PD-1 off. So it's a really interesting signal, and what that signal means and how we can improve this is really, I think, important going forward. And then the other question is, as Gina also suggested, this idea of the B cell follicles and why we think it's important. And this is a study that, Yoshi, who I think is now you guys led many years ago, and hopefully you're writing this up now. But in this original paper, one of the major observations that Yoshi found was that, in animals that have lead controllers, so they have really strong control of the virus, where to the point where they actually can really effectively prevent, limit our RNA expression in many areas of the lymph node, but except where you see the B cell follicles. And within the B cell follicles is where we found most of the viral RNA expression in the lymph nodes. We don't see that so much in the progressors. Obviously, their T cells are not effective, so you see RNA expressed both in the follicles and outside the follicles. But if you are a lead controller, you have very low levels of plasma viral loads. Most of the viral expression is only restricted within the follicles, which then began to lead us to suggest that, the follicles are really important because they are immune privileged site. It's very hard for you to control RNA expression there. And you have to think, if you stop ART and you can induce control in many different sites, even after ART, this might be an important site where most of the virus is rebounding from, because the T cells aren't effective at controlling virus within that site. So that was really what led us to continue this idea of maybe, one of the things that is driving this, one of the things that is leading us to not really getting a good handle of why the CD8 T cells are not controlling very well is because, a lot of the rebounding virus is coming from the follicles. And if we can disrupt those follicles, perhaps it will allow us to understand that maybe the goal is to target the follicles and we have to drive responses into the follicles. And if we do that, maybe that will be a way of slowing down rebounding viremia. So, there isn't really a good way of getting rid of follicles. Follicles are there for a reason. Most of them are full of B cells. And so one of the ways we've tried to do this is by giving anti-CD20, which is initial studies were done with rituximab, which is a human anti-CD20 antibody, but now we have resized versions of that antibody. In some very preliminary a lot of preliminary studies we've done, Yoshi back in the day was looking at lead controllers, and when he gave them B cell depletion with the anti-CD20 antibody, he showed that, if you B cell deplete elite controllers, that actually further induces control of the virus. So it really was pointing to the fact that, the follicles were important in the context of elite control, because the elite controllers were able to control virus many different sites, but when you disrupt the follicles, you can actually further improve viral control. So we wanted to ask this question in the context of post-ART release. We infect monkeys, put them on ART for a long time, and then begin to ask the question at the time of rebound, deplete B cells, and then look at what happens to the dynamics of reactivation. And just to give us an idea whether towards the importance or the role of the B cell follicles in this early viral rebound dynamics. And so, in this initial study, what we did is we gave, CD20 2 doses before ATR and continued 2 doses after. And, we got really good depletion of B cells in the blood. And we said we got good depletion of the B cells in the follicles, but in the lymph nodes, but it wasn't complete. For the most part, what we see is that in some animals, you still have, follicular structures present, but they're much smaller. But also one of the things that BCD20 does is actually, allows a little bit more CD8 T cells to infiltrate within the follicles. So if you look at the staining, which I don't have time to show here, but we see more CD8 T cells, that we can see visible within the follicular area. So we think it's really a good strategy to at least to disrupt the follicles, not to probably completely eliminate them. Anyway, so in this study, actually, we had animals, we had a protective MHC allele and no protective MHC allele. And then we looked at the dynamics of viral replication after ART release when you have a protective MHC allele and you don't. And so when you have a protective MHC allele and we see, again, we didn't really see a difference in time to rebound. So that initial ramp up of viremia after we stop ART was not really altered with the follicles. However, what we did see again was this early improvement in post-ART viral control. So, and this was more pronounced in animals that had a protective MHC allele. So disrupting the B cells at the time of ART, if you are predisposed to have very good cytotoxic T cells, can further enhance viral control. And actually we see here. So remember, the animals in black also have a protective MHC allele, but they had their B cells intact. Here, the B cells were depleted. At least their follicles were depleted here. And we can actually see that we get better control. So it does give us an indication, the B cell follicles are important. They probably are not important in driving this early rebound dynamics, but they do seem to be important when it comes to, at least if you have a good T cell response, when it comes to, this initial decline from peak. So if you have no follicles, then the T cells actually can drive down viremia much better than when you have your follicles intact. But the virus is always trying to fight back, and so you have the virus replicating again. But then we seem to show that over time you get better control overall if you have depleted your B cells, at least in this cohort at the time of ART. We didn't see as much of a risk difference if you have no protective MHC allele, so you are not really predisposed to generate a very good cytotoxic T cell response. Although the trend seems to show that the viral loads were slightly lower, but it wasn't necessarily significant by it too. So at least it tells us, at least in this study, that B cell follicles, they help, but they don't help completely. But then again, as I said, we're always looking for signals, and so it tells us that at least it's important to make sure you have good overall control first before you then begin to add on the role the follicles may be. So the follicles may be important, but what's more important is to really get a handle of getting very good cytotoxic T cells to control rebounding viremia. And so we followed up this study again. We're asking the question of whether if we combine this with IL-15. And so in this context, what we did is that we got 3 groups of animals that were all BO8. So BO8 is an MHC allele that is protective, there's very enriched in controllers in an NHP model when they're infected with SIV239. And so we got 3 groups of them. And what we did is that we now gave them B cell depletion through ATI. But prior to B cell depletion, we also gave them this molecule called Na20. Now, Na20 is a fusion molecule of the IL-15 superagonist Na203, which is currently in clinical trials. It's being used quite a lot now for HIV and HIV cure interventions. And then this molecule is fused to rituximab, which is the human CD20 antibody. So the idea of fusing this fusion molecule is that it will stimulate NK cells to drive enhance ADCC to probably then enhance control of the virus when to enhance elimination of CD8 B cells. So I think it was originally designed to better deplete B cells in the tissues, but we were also interested in using it to actually use IL-15 to enhance and expand cytotoxic T cells. And there was data that was published by Jonas Sacha's group showing that Na203 given to monkeys can actually drive CD8 T cells into the follicles. And George Pavakis also has shown with his heterodimeric IL-15 that given IL-15 to monkeys can actually drive SIV-specific T cells into the lymph nodes and potentially even into follicles. So, it would seem like a win-win where we would give IL-15 to do 2 things. One, this molecule can deplete B cells, but then it will also expand and activate CD8 T cells and NK cells. And so we decided, and then obviously with all these experiments, timing is everything. When do we give it? These are all the questions, right? So in this context, we designed a study where we gave it either during full ART suppression and then maintain our then we gave 3 doses of Na20, and then we maintain CD20 long term. And then in the second group, we said, well, maybe we need to start this at the time of ATI. And so we gave 3 doses just at the time of ATI, and then we continued with CD20 because we wanted to make sure we were maintaining steady state We wanted to make sure we were maintaining steady state CD20 depletion. And then we had a control group. So what did we see? I mean, so in a nutshell, the YADN molecule is very effective at depleting B cells, both in the blood and in the tissues. But one of the things I wanted to highlight was that when you give IL-15, you do actually get an activation and increased proliferation of CD4 memory T cells. So here, this was the period we gave the N820. You can see proliferation of CD4 memory T cells. And in the group that started N820 at the time of ART, we also again see proliferation of CD4 memory T cells. So what happened in terms of when we looked at post-ART viral loads, again, we didn't see a difference in this initial time to viral rebound. This seemed to be very different very similar. Even the upswing of the virus was very similar. The only thing is that in the animals that got IL-15 around this time, that started IL-15 around the time of ATI, you can actually see that was where we activated CD4 cells. They also had increased viremia, but IL-15 also potentially also activated CD8 T cells, and so even though they had got increased viremia, this was controlled very rapidly. And we see some lower plasma viral loads, but this was not really significant between the 3 groups. So really it tells us that maybe giving too much IL-15 potentially around this time might be an issue. It's questions we're trying to figure out going forward. But at least it's telling us that for the most part, that just depleting the follicles and just combining that with IL-15 potentially was not enough to show us that we can drive early or post-ART control. So the last thing I'm going to focus on right now is the question that we then wanted to ask, which was, well, maybe what we just need to do is increase the number of CD antigen-specific T cells. And the best way to do this is using a therapeutic vaccine, and particularly one that is targeted at enhancing CD8 T cell responses. Not only did we want to vaccinate animals with a therapeutic vaccine, but most of the time when vaccinations are done during full suppressive ART, and then when the cells are at rest, they wait after maybe a month or several weeks after vaccination, and then look at and then stop ART and actually ask the question of what happens to the T cells. But I think we wanted to take a different approach here because of the idea of, we wanted to see whether the reason why we're not getting an earlier intercept of the virus is because there are just not enough CD8 T cells that are activated and ready to respond to the virus at the time of ART release. And so, our strategy was to ask, well, not only do we want to therapeutically vaccinate to increase the pool of antigen-specific T cells, but also just prior to ATI, we want to be able to boost those cells and then stop ART. And then ask the question whether, boosting cells prior to ART can delay rebound and potentially can that drive post-ART control. And so to do this, we partnered with CureVac, who have developed this mRNA vaccine construct. This construct was specifically using it was only designed to target T cells, and so we only included full-length SIV GAG because we only wanted to generate GAG-specific T cell responses. It's an unmodified mRNA, so it's slightly different from the current COVID vaccines that we used in the clinic, which are all based on the modified mRNA platform that Drew Weissman developed a long time ago. This is a non-modified nucleic mRNA, which are much better at generating very robust T cell responses. So that's the difference in the vectors. It's also encapsulated in lipid nanoparticles, and it's usually administered intramuscularly. And we've done a lot of preliminary data. In fact, this is the 3rd generation of the vector we've tested. 1st and 2nd generation were given in different routes, different administrations, but they're all very immunogenic. And this actually this 3rd generation is much more immunogenic than some of the previous ones, and we were able to get really high specific T cell responses in tissues in particular, which is where we really were targeting, not just in the blood. We wanted to make sure that we can get good high specific T cells in the tissues. So this was our study design. As I said, animals were infected, they were put on ART, and then during ART, we began our vaccination cycle, which included 3 doses of the vaccine. 3 weeks apart, and then we do a single boost 20 weeks after. And then again, just prior to ATI, we gave a final boost. And the question is that, a boost just prior to ATI, will that be enough to enhance, an earlier intercept of the rebounding virus? This showing that, the animals prior to during infection and during out suppression were fully suppressed, and there was really no difference in terms of either the peak plasma viral loads or the total viremia that we were able to detect between the two groups of animals. And as I said, the vaccination included 5 intramuscular injections at 100 microgram each at these different intervals. We also had a control vector, so it wasn't untreated control. The control vector was expressing the rabies virus glycoprotein. And the last vaccination was given just 2 weeks prior to ART. And we had specifically designed it that way because, we had all the optimization studies we had done, we knew exactly when we were expecting the responses to peak. And so we wanted ART interruption to occur at the time when we were expecting the T-cell responses to peak post-vaccination. This is just showing that the vaccine is very immunogenic. It generates high levels of GAG-specific T-cells. So this is in the peripheral blood, where you can see, compared to the control vaccinated animals, we see high frequencies every time, either following the initial 3 doses and then the boosts at week 20. When we look at the lung wash, which is the bowel, which is, an indication of, T-cells within the mucosal compartment, we see really high frequencies of GAG-specific responses in the bowel in response to we also see CD4 responses, but they were not as high as the CD8 T-cell responses, which is kind of what we were hoping for, because we don't want too much CD4 stimulation, although we just want just enough to facilitate CD4 help, but we want really more of CD8 T-cells. We also were able to see increase of GAG-specific CD8 T-cells in the lymph node as well post-vaccination. Some of the animals in each group had tetramers, so we were able to look at, because some of the animals were AO1, so it allowed us to look at tetramer responses. So we could use tetramers to look at GAG-CM9 specific T cells, as well as TAT-SL8. So TAT-SL8 is also an immunodominant epitope for MAMO-AO1, but it's not included in the vaccine, and we can use that as an internal control, and we don't see any increase in TAT-SL8, but we only see increases in GAG-CM9. And, we looked across several different tissues, and that's kind of the benefit, one of the advantages of the non-human primate models that allows us to do these really intense biopsies longitudinally. So we're able to look at peripheral lymph nodes, mesenteric lymph nodes, spleen biopsies, bone marrow. We even did liver biopsies, small intestine, rectum, in addition to the bowel, to just look at these GAG tetramer-specific responses, and we can actually say the vaccination across several different tissues the vaccine was able to increase GAG-specific T cells across several different tissues. And so, as I said, the main idea here was not only to vaccinate during full ART, but just also to time the final vaccination, the final boost at the time of ART release, so that the idea is, as I said, can we increase increase the number of SIV-specific T cells that can respond to the reactivating virus at the time the ART is being released. So, and I think for the most part we achieved that. We vaccinated 2 weeks. The last boost was 2 weeks prior to ART. We can see in the blood the increase of GAG-specific CD8 T cells in the blood occurring at the time ART was being released. And we saw even higher frequencies in the lung at the time of ART release. This is in contrast to what happens during natural infection. So the green is the control animals, where we stop ART and then begin to see a gradual increase in CD8 T cells in the blood. And then, also a gradual increase in CD8 T cells in the bowel, which as shows that the CD8 T cell expansion is occurring after viremia and not before. So 2 days prior to ART release, we did biopsies again and looked across several tissues. And then we also repeated those biopsies by day 18 after ART release, just to show that the T cells were also not only expanded across different tissues, but they were maintained at least through the first 2 weeks of ART release. And this is done by ICS. And so then we looked at cell-associated viral loads again. So here I'm just showing that there was no difference in cell-associated viral loads during the first pre-ART release. And then, but at day 18, we see a significant increase in RNA expression in the controls, showing that, at least the CD8 T cells, the vaccine has elicited some response to actually limit CD8 T cells at SIV RNA expression. We see that in the lymph nodes and the mesenteric as well. Though we don't really see much change in DNA, but at least we should show that, there is restriction of RNA by the vaccine. And finally, what we've been trying to do all this while is to look at time to rebound, and we're showing that, time to rebound is significantly delayed. So the median time to rebound without vaccination was about 11 days. But when you have a vaccine on board, the mean time to rebound was about 20 days. And then this is just highlighting, overlaying the T cell responses with rebounding viremia. So as we increase the T cell responses, we see a delay in time to rebound. But as the T cell responses begin to wane, both in the bowel and the lung, is when we begin to see rebound occurring. And these responses begin to wane over time. This is in contrast to what happens, during natural infection, which I said T cell responses follow rebounding viremia; they don't precede it. And so this is kind of overall, we see not only a delay in time to rebound, but we see early control over the first couple of weeks, that these T cell responses are maintained over the first 6 weeks, at least 6 to 7 weeks, we see better control of the virus, but over time, this is not sustained. And so, the questions we're trying to answer is how can we sustain this response long term? And I notice we're almost at time, so I would very briefly say that, one of the things we were trying to see to ask the question, can we sustain it, is to actually then increase the number of antigens we use. So we designed another study where we added NF and POL to the vaccine. And, I won't have time to go into all the details. It was immunogenic. And also, again, it increased the responses to these three. And we were able to delay time to rebound. But one of the things we did see is that in this context, when we added more inserts, we didn't see as much of an improvement in viral control. There was a bit of more of a delay in terms of the initial upswing of the virus, but the overall control was not really different when you added more inserts. And so we went back and compared what happens when you add to gag responses, where you add all 3 inserts into the vector versus if you have gag alone. And you can see very clearly by adding more inserts, it decreases the responses, which is something we're trying to understand why that happens by adding more. We were hoping to increase breadth, but when you try to increase breadth, it's almost as if you deflect the immune system and it's not targeting the right thing. And so, we decrease the responses, and we can actually see that, the gag alone actually gives you a much, the gag alone somehow gives you better control early on. And then when we look, and this is important because when we looked at all the correlates, and we looked at many different things looking for correlates, one of the main correlates we saw was the amount of gag-specific responses in the tissues, especially in the bowel, was the main correlate that led to durable post-hoc control. So the animals that had higher gag-specific CD8 T cells in the bowel at the time of ART release had better post-hoc viral control. So they had lower viral loads at set point. So we know that one of the things that seems to drive better control is just increasing those numbers, particularly within the tissues. And so we're still moving forward. So if most of the animals that had the trivalent had lower responses because we deflected. And so they actually had higher set points relative to the animals that had gag by itself. All right, so I'm just going to summarize because I think we're at time, but just to say that, we know that, therapeutic vaccination is a really good strategy to be able to drive, to potentially drive post-hoc viral control. This vaccination strategy that we showed here induces really high frequencies of T cell responses. And if you give a boost just prior to ATI, we can actually improve the delay time to rebound, but and improve the intercept of rebounding infections. But, this was not really enough to facilitate more durable control. So, a lot of ongoing studies are really focused on exploring strategies to further enhance the efficacy of a T cell targeted vaccination strategy to induce remission. And I just want to thank everybody in my group and my mentors, Lewis and Jeff, and funding and Dare and everybody who has made this possible. And I will stop there. Thank you. I'm happy to take any questions.
That's great, Afam. I know we're at time, but if you can stay on for a few minutes, I'm sure there are probably a few who can stay on for some questions. I can start with one of is a pretty hopeful end there that a therapeutic vaccine at least shows some sign of delaying rebound when timed correctly, right? right at the moment before the virus starts coming back. And I'm wondering if you can sort of what the next step is to extend that protection, and maybe linking in with Gina's, talk, focusing on, sort of maintaining that proliferative capacity of the CD8s long term as opposed to just sort of the transient protection. What do you think is the next step to extend the signal that you got there?
Yeah, I mean, those are things that, keep me up at night thinking about all different things I'm thinking about. I mean, as I said, one of the things we see that, PD-1 by itself actually does do something. And it would be an intriguing question to see if we, depending on how we can time the vaccine, give the vaccination and PD-1 at the time when we were expecting the T cells to wane, whether we can sustain that proliferative response by giving PD-1 at that time. And so that would be one strategy. We're also interested in this idea of TCF1 and the role it plays in upregulating or sustaining the prolific capacity or the pool of cells that can proliferate. And so we have we started by Lydie before, and I inherited her group, and so we've been working on different GSK3 inhibitors, which actually are very potent at upregulating TCF1, and we have found a few that work really well. But the question is, it's great to get things that work in vitro, but how they work in vivo, how we will apply it in vivo, and how we will apply it so that we can increase the pool of highly proliferative T cells in combination with a vaccine is what we're trying to understand. So I think there are potential strategies to do that, and we're trying to think about them. Great.
Yeah, questions from others?
There's one in the chat, Peter.
Oh, Yoshi, your old pal, Afam.
Yeah, I see.
Who we were delighted to recruit here at UCSF.
Yeah.
Well, thank you very much. Great talk and introducing my didn't work project. So yeah, but the PD-1 and also messenger RNA vaccine very impressive. And I was curious, how was the PD-1 treatment preferentially enhance the CD8 response? And also, if you remember, did the treatment also change the some CD4 response, or I mean, like animal who has a high viremia tended to express high PD-1, right? Extreme high, but the antibody treatment kind of block PD-1 expression. It means it may block the indirect cell death or whatever. So.
Yeah, great question. So we didn't see a significant increase in the overall antigen-specific T cell responses when we gave PD-1. I don't think it overall increases it. So I think it may be an increase in functionality and not magnitude, because all the cells proliferate, right? So you would expect the proliferation of both infected antigen-specific and non-antigen-specific cells. We also see CD4 proliferation happening, and whether that also is a yin and yang thing, right? You're driving more proliferation of CD4 cells, you're also activating it and increasing the virus, and then having to have more work for the T cells to can catch up. For the most part, we see proliferation of CD4 cells. I don't think we see any changes in the antigen-specific CD4 cells, other than all of them seem to increase and proliferate in response to PD-1. If that answers your question, but yeah, I think that there is an issue with these agents that can stimulate CD4 cells because they may favor, to a certain degree, increasing more of the virus. And so, how we use them safely and understand how we use them is one of the things we're thinking about.
Okay, thank you.
And Steve, you have a question?
Oh yeah, no, yeah. So in the clinic right now, in the cure studies that are happening at UCSF and Denmark and Rockefeller, UK, and so forth, and there's these very consistent stories coming out now that broadly neutralizing antibodies can alter post-ART dynamics, leading to control, at least in some people. But they don't do anything in monkeys so far. And I'm wondering why.
So far, I don't know if I would say we don't see anything in monkeys. I said we haven't observed it so far, especially with SIV239. And you also remember that we don't have a lot of neutralizing antibodies against 239 that have been tested yet. I'm hoping that more are being developed and people are interested in developing more of them. So it's taken a long time before we've got all these antibodies that have moved into the clinic, and now they're showing that they're being effective. So remember too that in our studies we only gave a single dose at ART. We haven't done long-term dosing to see whether you see with long-term dosing you see an effect. We haven't done a lot of those studies, and also the Danish studies, as you were showing, they actually gave the BNAB at the time of ART initiation, right? And then they are showing that there was an effect long term. Some of those studies we have not done. I don't think a lot of those studies have been done in monkeys with the 239. So I would say that the field, it's still growing, and hopefully with time we'll still do more of those studies to ask some of these questions that are done. I think if we do them similarly in the monkey as it's done in human, then we can ask the question. But right now it's too early to ask. Our studies were not designed to answer that. Our studies were initially just designed to slow the virus down, and that's why we did it.
Okay. And so I know we're running late, but I have one burning question to ask to maybe close it out. And that's, you referred to these oscillation patterns that you often see in the post-rebound period. As the immune system is sort of, struggling to maintain control on the virus. What do you think explains those dynamics? And can you use the non-human primate model to try and understand what exactly is happening? In other words, is this immune escape, or is the virus traveling to different anatomic sites and reestablishing foci of infection and then exhaustion of existing cells? Do you have a sense of what may be going on?
Yeah, well, that's the predator versus prey thing. So, with the PD-1 study, actually, we did actually ask the question of whether, because the obvious question was, well, what's happening is you're just driving mutations. And so, Brandon actually, with the oscillations, he actually did some sequencing of different peaks. And, in some monkeys, yes, you see some mutations that were driving the oscillation, but in quite a few monkeys, it wasn't being driven by, spontaneous mutations. So, it wasn't enough to say, well, the virus was escaping, and that's why you were seeing the oscillations. And so what it really is, I don't know. It could just be the nature of the CD4 T cells as they're being controlled and driving our increased RNA, and then and as control happens, you get suppression, and then there's more. It could be influx and outflux of cells. It's still unclear, and obviously we're really keen to answer that, because it does seem like that's also something that's it's very similar in humans, and Steve has said that what happens in the kitchen sink study or so. So it's an interesting phenomenon that's across both species, and that would be nice to get a handle of that. But I don't know if I have a clear answer just yet.
Great. All right. And it looked like there was one trailing question in the chat about BNAbs and whether they can be compromised by mutations. I think that's a pretty straightforward question.
They can, and even the ones we've used so far, at least the ATS-103 it's easily escaped. Especially that's why we try and use a combination therapy. But yeah, I agree.
Yeah. Okay. Great. All right. Well, thanks, Afam, and thanks, Sheena, for great talks and a fantastic discussion. Thanks everyone for joining, and we'll see you again next month.
All right. Thank you. Thanks for inviting me. This was fun. Thank you. Bye.